From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 25th, 2023. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 9th episode of Matters Microbial. Thanks for listening and or watching. First, a denizen of my Wunderkammer. One of my microbial heroes is the late, great Carl Woese, who was a driving force behind a paradigm shift demonstrating that there are three domains of life: eukaryotes, bacteria, and the enigmatic archaea. Here is a needle-felted statue of Dr. Woese that I commissioned from Amy Wright many years ago. I have linked to the story of the sculpture in the show notes. That story and the example of Dr. Woese and his research journey is so important, I even put in a link to an article I published about Dr. Woese, as well as a link to an oldish video from the American Society of Microbiology. Dr. Woese was a remarkable scientist and well worth your time. As far as today's inspirational lapel pins, the first is a reminder to always be yourself. Authenticity matters in all things and cannot be faked. The second pin is a quote by the great Walt Whitman, "Be curious, not judgmental," which was featured as an integral part of a particular episode of the wonderful television series Ted Lasso. Again, these are lessons that I truly believe in and hold dear. The fourth week of my microbiology class has been a lot of microbial fun as my Micronauts explore. They've carried out single colony PCR reactions with their water bottle buddies. We will learn this week if the PCRs have been successful, and if so, send off the purified amplicons for sequencing to determine the possible identities of each of their isolates. We will also isolate more of our marine microbiological safari candidates to single colonies to process in a similar way in future weeks. These include, as you can see, agar degraders, some very odd colony morphologies, which fascinate me, and even something very strange, what looks like filamentous microbes in a marine sample. The real point here is that the Micronauts get to learn the identity of these denizens of the deep, which they isolated. While the most important tool for microbial scientists remains the brain, there are so many pieces of equipment necessary for a working scientist. Today, Sebastian Cocioba, a citizen scientist with endless energy and myriad interests, talks with us about his path in science, some of the tools he has made for others, including yours truly, and how he views science and scientists. Welcome to our Quality Quorum, Sebastian.
Hi, Mark. Thanks for having me.
It's really great to see you. So can I start the ball rolling, as it were, microbially speaking?
Sure, go for it.
So you've basically done several things for me, just in order for people to know the background. I first noticed you on social media as you were making different items for other scientists. Your interests are very broad-based, and I want people to know before they hear anything about your background, the kinds of things that you have done, not just for me, but for other scientists. So the first thing that you did is provided me with an LED-driven transilluminator that allows us to visualize DNA on agarose gels, as in the photograph that you're seeing now. And I understand that you got that idea from some other people that had a very highly rated or highly priced version of that also ones using types of UV light that are a little more intense than we should have around people.
Yeah, that's exactly correct. Yeah, the overwhelming necessity for safe transilluminators has brought me to one of the few companies that actually offered it at the time, using blue light instead of ultraviolet light, which is far safer. It doesn't require a face shield, won't tan your skin, right? But they were selling it at an egregious price. And snooping into their patents, I noticed that it's not really a protectable claim. So I said, let's go. Let's make an open source thing. And yeah, being able to just block out that blue light to allow the green shine of the intercalation dye to shine through was a really simple build. It's just LEDs, resistors, and a USB cable.
It is absolutely so useful to my students. We use it every time we run a gel. Now, there are transilluminators we have upstairs that can use a computer to estimate sizes of bands. But I tell you that to go upstairs and try to sign on to use it time-wise, it's so much easier to use the small device that you made through 3D printing and soldering a bunch of LEDs. And I cannot thank you enough for that. You can actually see the bands very clearly as I've ever seen them in very, very expensive transilluminators. So thank you for that.
My pleasure.
It's really amazing, and I'm really thankful for each piece of equipment. And who knows, more to come.
Definitely. There's a whole suite of things that on the horizon, because as I realized that the fancy tools that I've acquired over time don't need to be that fancy. I use them to vet the homemade things, right? So every single tool that I build, I always build with reference to a commercial unit that I have on standby to be able to modify and tweak and tune until the cheap bespoke stuff is just as good or good enough for most microbiological work. And it turns out you can do an enormous amount of replacing and revisiting to traditional academic apparatuses. The main drive for the bulk of the inventions that I make is necessity and borderline desperation. So I'm a self-funded biologist, right? So everything comes out of my pocket. And a lot of the tools to do basic research are simple in practice and expensive in price tag. And I've noticed that there's a tremendous markup for really simple instruments, and that got me thinking, I should probably just make this. The equivalent machine that I made for you was sold by a company who I'm not going to give clout to for about $600, and I would sell mine for about $64. So an order of magnitude undercut, and essentially at cost. And one of the main reasons is I'm trying to do right by who I was when I first started, like even more penniless than I am now, and like ambitious to get going, and I had very little help. And essentially, the biggest roadblock for me was access. So everything I tried to do is focused on how can I bring more people into the fold, and what kind of instruments we can design along the way, and make like a community of like self-sufficient biologists, ideally.
Well, you certainly have impressed the heck, not just out of my students, but some of the technicians that work in this building. They wanted me to extend this to you, that you're quite the solderist, which I did not know was a term, but I guess it is. Yeah. The second thing that happened, and I want to repeat kind of a comment that you made about the sense of community that exists. I don't want to get into the downside of social media, because that's a whole encyclopedia worth of verbiage. But there are also many good benefits of social media, and connecting people at great distance from one another is one of them. So for many years, I have been taking advantage of something that I could purchase commercially with various strains of E. coli carrying plasmids that expressed different pigments. And it's always good for students to learn how to handle microbes in that fashion. And then I noticed that you had an improved set of these things, and I want to emphasize that you did the constructions yourself. And this is not something that you bought from a company. This is stuff that you kind of designed, and I want to emphasize to the listeners what that means. For example, here is a Petri dish showing some of the palette of the different colors that can be used. In addition, Here is how it looks when it's grown in liquid media. I use these to help students learn how to handle microbes in a way that engages them. Can you tell us a little bit about the journey that created these strains?
Sure. So because I'm the captain of my tiny ship, I can steer it wherever I want it to go. And one of my big obsessions, this kind of like broad strokes obsession of the living world, is color, right? How color comes about, how biology expresses itself. And I find a lot of biological pigments really inspiring, and it forms this kind of need to collect these palettes to be able to interface with them. And so basically, I was really upset that a lot of the labs that I experienced while I was at university had just GFP, just green fluorescent protein, required a UV light to see. A lot of the times we had needed a face shield because it was shortwave UV light. And I asked, what about other colors? And the TA at the lab at the time told me I should look up Roger Tien. And that took me into this wonderful rabbit hole of mutagenesis and colors from the deep, and I was obsessed. And I tried to find like ways to purchase these. Is it available? And lo and behold, it was very difficult to actually find an answer to that. And most of these are for educational or academic, specifically academic use only. So this was about 12 years ago, where the concept of a home lab was like burgeoning, but not necessarily was there any type of places that would sell to the amateur biologist, or in many cases even to like the high school teacher. And so after like learning a whole bunch about how to handle DNA and how to basically cobble together things like very angry, cumbersome Legos, eventually I got to a point where I said, I'd like to have these colors and explore with them. And so I went on to GenBank. I cross-referenced a bunch of things on Uniprot, two of my favorite tools, to kind of search for things that have color specifically in daylight conditions. So fluorescence is beautiful, but I'm more interested in things that express in broad daylight. All right, so those are called chromoproteins. There's many fluorescent proteins that can be chromogenic, as in they absorb specific wavelengths of white daylight and reflect back different colors. But there's not a lot of insight into how these fluorescent proteins also produce these colors. Like, for example, my favorite blue protein happens to be a far red fluorescent protein that nowhere in that description has the word blue in it, right? So it just happens to be an interesting kind of quantum effect of the folding of those proteins. But yeah, so I thought, why not just have one color representative for the rainbow, hopefully, right? Like a Roy G. Biv type of thing. Got a little upset at indigo, right? Blue, indigo, and violet. It's not really that much of a distinction. And so I just went blue and purple, right? So I have red, orange, yellow, green, blue, purple. Those have been like vital tools in my personal research, using them as plant markers, using them as bacterial markers. But one day on Instagram, where I post my pretty stuff, somebody said, hey, I'll buy a set off of you. And I said, oh, sure. I never thought, well, selling plasmids, okay. So all the stuff that I work with is either wild type or distinctly open source. Like the Public Domain Gazette is a wonderful source of open source sequences that they basically they basically trawl the murky depths of patent expirations to find what is now in the public domain that could be useful for scientists. And so after a while, I cobbled together a nice open source kit, and I opened up an Etsy store because these were handmade with love in New York City, and I still hold by that to this day. Everything that I make, I do traditional cloning. It's all by hand. I have barely any automation. And so I find it a little bit of like an artisanal craft that I could share with people. And eventually a bunch of educators contacted me and said, hey, we've been using some of your strains. We really like them. Can we share them? And I said, of course. And eventually more people started coming to my shop. And eventually you got a set, which was really cool. Now it's mostly by word of mouth because Etsy decided to shut down my store, even though it's biosafety level 1 and in the US, shipping BSL-1 organisms and E. coli K12 is not regulated. But they didn't want to do the trouble of sorting that out, so they just kind of blocked my store. So now I'm building my own shop at atinygreencell.com, where I'm going to be hosting all my wares and focusing more on more colors, especially green. So the green I have is a green fluorescent protein, so it's not green in daylight, and that doesn't really vibe with the entire concept of, daylight absorbing chromoproteins. So I found out that if you take a yellow chromoprotein and a blue chromoprotein and you fuse them together, it makes green, which is amazing. That opens up a whole new possibility for novel colors that we can then immediately put in the public domain so that there's no weird IP issues, and make these kind of new open source tools and hopefully get students involved to start producing their own unique colors. Like imagine teaming up with Pantone for the color of the year and then having people compete to try to make the color of the year using fused chromoproteins. A little baby iGEM project.
I think this is so fascinating. And one of the things that you and I have talked about a little bit, and I don't want to divert the conversation too much, is I've used commercial sources of similar plasmids, though not quite with the chromaticity of yours, for lack of a better term. But what I will say is in using yours, I found these unusual mixed colonies that are very hard to explain. And it's funny when people look at them, they come up with, guesswork. But the important thing for me almost isn't how those things appear to have happened, but that the students noticed it. Because truthfully, the best piece of equipment in a laboratory is the one between the students' ears. And so I am super grateful for what you've done. The final thing that I'd like to say before I start grilling you on your fascinating background is the business of measuring optical density of bacteria. And so after back and forths over quite a period of time, and I don't have a set of data to share, Sebastian, you sent me what is, I guess for lack of a better term, could be called a turbidostat, which you greatly simplified for tiny minds like my own. Because when you start to look at how turbidostats and chemostats work, there's a certain degree of complexity and a certain degree of fine-tuning. And this, again, is something that you're particularly good at, in my view, is finding tools for the user, for lack of a better term. And so if you look at the photograph that's currently being shown, this is a turbidostat, or one of 8 channels that you made for me. I appreciate the googly eyes in particular. But I can't wait to be using it to measure actually fairly interesting differences in growth rate among some of the microbes that I use. And so I wanted to thank you that in advance, though I don't yet have data to share with you.
My pleasure. This is a dream come true to be able to build tools that are good enough to share.
So I won't even deal with what was the thought behind that one, because I'm sure you'll talk about the parent project that put off this progeny that you sent me. But the first question I'd like to ask you is a little bit about your journey. Because when people look into your background, they find out you're not at a university, nor do you amazingly have a PhD, which I would have assumed based on the way that you do your work and communicate. And this is all about the role of citizen science, which was such a big deal to me when I was growing up and kind of fell out of favor. And folks like you, I don't like the term biohacker, but you know what I mean, are starting to bring this back. So please paint us a picture.
Okay. So that's always a fun conversation to have because it's multifaceted, but essentially the bulk of my life has been around the interest of science. I didn't know at the time, but I was just deeply fascinated, especially by plants, just mostly aesthetically, to be honest. Like there are just so many shapes and sizes, and every time I'd ask my teachers, like, why are oak leaves like oak leaves and maple leaves like maple leaves? They'd be like, I don't know, just go away. And that's fine. Middle school, I'm not expecting a lot. Same thing happened during high school, right? Slightly more explanation, just a little bit. It's like, oh, homeotic genes. Now walk away. Cool. Go fast forward to university, and I was like 3 years into my undergraduate in general biology. Gave up on the concept of being a doctor. My grades were crap, and really got bit by the research bug. Right, so I had the enormous privilege of working in Vitaly Sitovsky's lab at Stony Brook University, who's one of the godfathers of agrobacterium research. And I didn't know at the time, But during the interview with him, he's like, "All right, you got 5 minutes. Why do you want to be here?" I'm just like, "I like plants." And he pauses it. He's like, "You're not a pre-med?" I said, "No." And they're like, "Oh, thank God." Okay. So next next question. What do you mean by you like plants? I'm like, "Well, I started doing orchid flasking, which is essentially taking orchid seed pods and putting them in media in like milk bottles and letting them germinate and then selling those progenies off. I basically used that to pay for a decent amount of my college education." And so I told him I had tissue culture skills, and he said, "Okay, you're starting tomorrow." And he threw some tomato engineering papers at me, and I'm just like, wow, okay, research time. And within the first couple of weeks, I was absolutely just head over heels for the concept of pulling knowledge out of the ether and then sharing it with people. Like that was a really unique high. And I've been chasing that ever since. But unfortunately, for family and financial reasons, I had to drop out of university. And but that research bug was still biting really, really hard. So I said, okay, time to level up my lab. And so I went from just like a simple micropropagation lab to more of like a basic research focus. And I had no real direction, but I had a lot of drive, and I just wanted to do science. Not the look cool holding a pipette science, but like really just try to explore the world around me. Maybe things that are new to me. It didn't have to be new to everybody. It didn't have to be the bleeding edge of knowledge, but it's just something that I could build my skills on at my own pace. Kind of like how an amateur home cook kind of explores old recipes and then kind of burns things at home trying to replicate Julia Child. That was a lot. I've burned a lot of soufflés for sure. And it took me about 15 years to get to where I am. And a lot of it was just kind of hitting my head against the wall, not knowing how to do anything, right? Because there wasn't really that big of a community back then. Luckily, I stumbled upon my now co-founder of Binomical Labs, Sungwon Lim, who basically taught me about restriction enzymes and how they work in terms of assembling plasmids. And eventually we just kind of teamed up and started focusing on research questions that we found interesting. He loves virology and phages. I'm interested in plants. And I'm just like, okay, no plant viruses in my lab, so we can't really mix together. But then microbiology is what really kind of sealed our fates in a way, in that we're now just obsessed with this. Like our very first large research paper was on sequencing of Deinococcus radiophilus, a microbe that's been around since 1962, but no one bothered to sequence it. And that was our first foray into doing an actual research project, right? So like a lot of expenditures on both our parts, doing nanopore sequencing. Christmas Day, 2 years into the project, we got scooped, which was heartbreaking, but also a wonderful thing to experience. And in terms of just like getting a feel for all the little nuances of being a scientist, working on a publication, getting scooped, pivoting, trying more, and diving deeper into the organism. And the best part was that the entire time we were free. We were free to explore whatever research questions we wanted. And I found that incredibly unique, where formal academics would need to do grantable research. We could just ask whatever question our pocket money could float us. And in doing so, you realize that there is an enormity of questions that have yet to be asked. And there was a paper from Nature, I believe 2016, that said just for eukaryotes, there's like 2.87 million species on the planet, and over 80% have yet to be discovered. Right? They show up in metagenomic polls, they show up in all sorts of places, and they line up with nothing that we know of so far. And that speaks volumes to the like nigh infinite amount of prokaryotes that have yet to be discovered. And so this sense of being born at the right time to be able to explore the world around you in a fairly affordable fashion has been just such an enormous driving force, to the point where I want to be able to spread this concept. I want more people to be able to do this if they so choose. And working within like the legal framework, because in many places bioengineering is definitely not allowed. In the US, it's an interesting BSL-1 gray area. And but you don't have to do bioengineering in order to do good science. There's plenty of just classical microbiology you can do, sequencing. I mean, if someone says you can't sequence, I would start questioning those specific politicians who decided that that's a thing. Not to put on my slightly radical hat, but yeah, there's just so much to explore, and you don't need a lot of money to be able to do this, right? And so I realized that because I lacked a lot of infrastructure starting, like during the beginnings of my journey, I became fairly good at building tools, and so now my entire like life's focus is building tools so that people can explore, and it's been incredibly rewarding. I'm a little bit jealous because some of the people use my tools to do very cool things, and I'm stuck cobbling, and that's fine. But there's a unique joy in being able to cobble something together in a couple days' time, and then the other person unlocks a new research path for themselves, be it a grad student, a PI, or just an explorer in their home lab. So it's been very special.
To me, it seems as if you're an interesting mixture between a electronic engineer and a molecular biologist in a lot of ways. And I'm sure that you might respond as, "Oh, anyone can do this," but I'm here to say that that's not really true in my knowledge. Perhaps I need to get out more. That might be a response that you want to make. But there are all kinds of things that I've wanted to do, and every time that I've spoken with you, you're like a pinwheel of ideas of what's possible and not. And I don't mean that you agree with everything. What I mean is that you have a good practical sense of what's possible. And
Appreciate that.
I am going to be leaning like heck on you later because, of course, in a few years, when I'm no longer affiliated with an institution, I want to look at questions that interest me. Does that sound like someone you know?
That is one of the more exciting prospects of the near future. That I would be so honored to be able to help outfit your lab and get you on the road to doing really cool research from the comfort of your home. Like
Oh, yeah. Yeah.
of all the things, having the classical microbiology experience and then being free, that is so special. So yeah, I'm first of all huge fan of your work, but more importantly, I'm excited for what you will do, not just what you have done, and hopefully use you as a guinea pig so that more people can join the fray.
It
'Cause
was
it's
very
so
interesting.
exciting.
Oh, that's very kind of you to say. It was so interesting the other day. I was talking with some of my students who were at my institution. There's one microbiology class, it's the one that I teach. And people are kind of introduced to a little bit of microbiology in earlier courses, but not really. So I have to make sure that they have the basic skills. And I thought of an idea that I tried this year, and it kind of reminded me of you in a way, in that I had students who didn't appreciate how many cells were in a single colony. And so I was able to find these little devices that one uses to take puncture biopsies, and they're sterile. And I had them punch a colony out, resuspend it in media, and do a dilution series to find out how many CFUs, cells there were per colony. And what was interesting is the students then said to me, does it depend on the size of the colony? Does it depend on the type of organism? And what I like is how kind of low tech the result is, but how it really dealt with the itch that a student had with a question.
Yeah, that's enormous. Like being able to have these like self-prompted questions that they could answer within like a couple classes. Just doing that dilution could give them just a sense of scale. That one little dot contains multitudes isn't just romantic poetry; it's truth. And I more of this, please. Especially outside of an academic setting where people maybe are homeschooled or things like
Mm.
that. Like just being able to experience this like edutainment of sorts. Not saying like producing content, but just like learning for the fun of it. There's so much of that, and there's so much that you can discover that's brand new, right? And so like having a pipeline or a vehicle for being able to like explore that, but then also document it and archive it such that others can find it indexably. That's something that me and my co-founder Sung have been working on for quite some time now, trying to like make that pipeline from curious at-home scientist to publication grade work that doesn't have to be formatted in an academic manner, but is still indexable, searchable, and archived until ideally the heat death of the universe. So yeah, it's been such an interesting struggle to figure out how to make that happen, but I definitely want more people to share their work.
I mean, take a step back for a moment. When Leeuwenhoek used his little lenses to look at pond water for the first time, he saw things that nobody else did. He was basically a tailor. Well, he's a cloth maker more than anything else, but he had the advantage of some resources to do those things. So he was scratching that own itch of wanting to know something more about the world around him. And too many times that gets beaten out of us in education in general. And I'm all for that kind of enthusiasm and that question, how do you know what you know?
Yeah, definitely. That Mark Twain quote, "Don't let your schooling get in the way of your education," is so powerful. And I'm
Yeah.
I'm not anti-school by no means, but like when people ask me, it's like, "Hey, how can I do what you do?" And I'm like, first, please stay in school, get your damn degree. It's so hard with that one. It's so hard. But the key take home is just like education comes in all forms, right?
Right.
And be able to experience, because so much of our brain is our visual cortex. We're visual learners. We have to experience things, and surprisingly, I'm a very slow learner, and I need to do stuff. I'm an experimentalist, not a theorist. So if you tell me the theory of PCR, it won't stick until I actually do it and fail. And I think a lot of people are like that as well. So trying to figure out how to get these type of experiments is really important.
Well, it's just fantastic. I'm watching our time carefully, and I wanted you to kind of paint a picture of what you would like to be doing, say, in 5 or 10 years, because I'll bet you have some cool ideas.
Yeah. So if I ever grow up, knock on wood, I'd like to be a flower designer. That's my ultimate goal. So like not necessarily take off the biologist hat, but maybe paint it a different color. So I want to design flowers that people haven't seen before, and then bring students into the fold with designing flowers as a way for them to learn about the natural world. And so this like mix between education and bio art, with a lot of like whimsy and a little bit of fantasy mixed in, is something where I'd like to lean towards. Now, I call my trajectory an uncareer because it's not about a job. Like I'll flip burgers if I can do science. I don't care. But in terms of what I want to do within the next 5 years, is definitely lean heavily towards more flower design. I felt like the first 15 years of my own career has been learning the tools, building the tools, and getting familiar with the edges of this pond that I'm in. And now I want to dive in. So like I've accrued enough knowledge and tools to be able to like seriously start doing morphological research, which has been like the big scary brick wall for me, because there's just so many moving parts, and you tweak a master regulator and the plant just kind of dies. So there's a tremendous amount of care that you need to take. And it's kind of like being a watchmaker, right? All those gears need to fit, and there's only so much room. But then you zoom out a little bit and you realize there's actually way more room than you think, because there's these wonderful things called gall gnats, right? That just lay eggs onto trees, and the galls that form, because there's hotly debated as to how that works, but one of the theories is that the larvae or the eggs, as they develop, start secreting plant hormones, different ratios over time, and it begets these crazy new morphologies of like star shapes and hairs and reds, yellows and blues, things that you don't see on like an oak leaf, for example. So there's untapped morphological potential in all of these plants. And so I've had these blinders on, thinking that homeotic genes that can control the shape of leaf, like the body plan, the ultimate cell fate of cells within a plant, are limited to the plant's genome. And they are not. So there's this new like hyperspace that I want to explore, just taking a cue from the weird little insects that happen to make these astounding cathedrals to biology.
It is amazing, especially when you think about things like agrobacterium, which we won't even get into, one of my favorite organisms. But are you aware of what leaf miners are?
Yes, yes. I've sadly had them 2 weeks ago, 3 weeks ago. Yeah.
So a leaf miner, for the listener and viewer, is a type of it's a caterpillar, a teeny caterpillar, and it lives within the actual width of a leaf where it's feeding on the plant. And there was a study from a few years back that I really adore telling students, and perhaps you already know it. Uh, it was found with I guess it's the apple leaf miner in France, that during the fall, when the apple trees would change color, there'd be these green spots on the otherwise yellow leaves. And they were surrounding where the leaf minders were. And it turns out that what's going on is those leaf miners contain a bacterium that you and I know of as Wolbachia. And this Wolbachia is forcing the cells that harbor it to overproduce cytokinin, a plant hormone, which is delaying senescence in the leaf, which allows more photosynthate to be consumed by the teeny tiny caterpillar.
So
And
cool.
I'd be happy to send you a reference to that paper if you're unaware of it. It's
Yeah, I'd love
something
to.
that's remarkable.
Yeah, the way insects interact has been really underrated, and we're just at the point where the tools are accessible enough to really start exploring these otherwise just wild and insane morphologies that you see underneath a leaf. It's hard to focus on just the road in front of you when you see all the trees and stuff on the floor. It's incredible. It really is.
And it's interesting because we're reductionist by nature. We like to look at one organism or one gene at a time, and that's been productive, except neither of those things are true in the natural world. Many genes working together, many organisms working together, but it's challenging to tease that apart in a way that's studyable. But nowadays, we're getting closer to it. I hesitate to call it systems biology because there are as many definitions as there are systems biologists, I think.
Yeah.
But I absolutely believe that there's so much to discover and there's a place for almost everyone in it. And you've done a wonderful job showing a route that I think will become increasingly important in the future.
That means a ton. Thank you so much.
And normally, what I do, and this doesn't apply to you, because truthfully, you're not a microbiologist, you are someone who uses microbes.
Hmm.
So I often ask people who are microbiologists, what is the one thing that sold them on becoming a microbiologist? But for you, I will say, what is the one experience that sold you on being the kind of scientist that you are?
In in everything that I do, microbiology is the largest component, right? And in fact, when I try to teach like plant engineering to folks who aren't familiar, the first thing I tell them is like, okay, have you ever struck out bacteria? Have you grown them? And they're like, what does that have to do with plants? So like everything. If you want to interface with plants, you need to use microbes, and that was a very humbling experience because I thought as a plant bioengineer, whatever that means, I would be working with plants and chemicals and means of introducing DNA into the plants in a more abiotic fashion, like a gene gun, for example. And lo and behold, agrobacterium, it's just the greatest thing ever, and it can transform most things. And that concept of this like interplay where you want to make a plant, but to do that you have to understand enough microbiology, practical microbiology, to get to that point, is it just shows how interconnected everything is, and it's a wonderful teaching moment to say there's an interdisciplinary skill set that's required in order for you to do even the most quote unquote simplest things in plant engineering. Right, like the hello world of transforming Arabidopsis via floral dip. You're dipping it into something. That something is agrobacterium that needs to be prepped and groomed and excited and chummed and angry. It needs to be angry, and quantifying that and following these kind of protocols dogmatically have really only led me so far until I started leaning into how to measure these microbes more and how to know when are the angry est, or when are they the right amount of excited and virulent to be able to transfer genes. So an enormity of microbiology is a part of my daily life, but also it's this is a little bit more of a kind of like a guilty pleasure of sorts, but it's the short-term rapid prototyping of microbiology that gets me through the extremely long, tedious 8-month experiments in plants. Like the small experiments with media studies and stuff, just to give me that hit of dopamine while I'm waiting for my brown plants to maybe turn green in a petri dish, hoping that it took the gene I wanted. So there's a lot of time scales that microbiology likes to play on, where there's short-term gains, long-term gains, and infinite stimuli to keep me going, right? Especially since I burn the midnight oil essentially for myself and not necessarily getting paid for those things. That justifying the failures is sometimes really difficult to keep going. But just pivoting a little bit towards microbiology, it's been a refuge in the open ocean that is plant engineering. Microbiology has definitely been a welcomed small island along the way.
Well, that's why you're part of the quality quorum when you come right down to it.
Appreciate that.
Do you have any last bits of advice or things you would like to say to our listeners and or viewers?
I mean, biggest form of advice would just be explore your nature, whatever's around you. Like there's probably microbes on your shoe right now that could be industrially viable, or could open up a whole new way of editing DNA. I mean, there's stuff discovered every day that comes from like obscure places. In fact, wasn't it a grad student on a hike that discovered a new branch on the tree of life? That was insane. There's more and more tools accessible to us to be able to explore that. So like now is probably the best time ever to be able to get into like little homebrew microbiology exploration in whatever domain you'd like to work in. Big shout outs to Archaea though; they're incredible. So it's the probably the funnest like area in research in terms of microbiology for me. That is just learning just how wacky all of these archaeal cells are, and how many we're missing because they can't be cultured on solid media. Right, which is a nice little teaser for folks that want to explore with liquid culture stuff.
Well, you just wait a little bit. Ken Stedman will be giving a visit here. We're going to be having him on the show with the title Viruses from Hell, which I think
Cool. Very cool.
It should be starring some 80s action star. Actually, I did want to say parenthetically that the person you mentioned was a undergraduate student working for Tom Brock when he was at Indiana University. And Tom Brock was interested in learning if there were microbes that lived in hot springs in Yellowstone. And he and that undergraduate student described it in the late 1960s, the organism Thermus aquaticus, which was necessary for PCR to originally take place as an industry. Keep in mind that undergraduate's name, and boy, I wish I had a cool name like this, Hudson Freeze. It's like a supervillain's name. He's a fine cancer biologist in San Diego. So I would like to thank you very much for coming on Matters Microbial and chatting with myself a little while. It's been, as always, super exciting and therapeutic for me, and I hope our listeners.
Thank you so much for having me. This was really fun.
I'll be talking to you again soon.
Take care, Mark.
You bet. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes with fascinating links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Sebastian Cocioba can be found online at Binomica Labs. Many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum today. See you next week on Matters Microbial.